also like to pay respect to the elders, both past and present, and extend that respect to other Aboriginal and Torres Strait Islanders here today. So today's speaker, Jason, many of you know, he came to Peter and I 3.5 years ago wanting to do a PhD because he'd been doing honours with Matt and Jackie Wills over at Monash, and he'd been working on Sax, and he decided that if he really wanted to understand protein structures, he should learn crystallography. So he came over. And sorry, James, if you're in the audience, by the way. Anyway, and so as soon as he walked in the door, we realised he was a very enthusiastic and excitable kind of guy, and so we were very happy to take him on board and offered him a project trying to understand the way one of the pro-apoptotic proteins, Bak, oligomerizes and gets activated during apoptosis in order to understand the structural details of that process. And Jason took his typical mixture of enthusiasm and vigour and laid-back nature as well, and combined all that to be incredibly successful. And he's had a really good PhD, solved a bunch of different structures. He's had 2 published in Molecular Cell last year. You'll hear a bit about those today. A further 2 he'll talk to you a bit about today, which are yet to be published, but we're very excited about. And in addition to that, he's had a whole lot of, some of that work has initiated a drug discovery program, which we hope to really go headlong into now, which might be able to find small drug-like molecules which can inhibit apoptosis. Now, in addition to his great scientific work, he's also been he's got a very good social conscience, and he's been very, very proactive in, particularly in Aboriginal areas, and he's worked on the Weehai Reconciliation Committee for the last 1.5 years or so. And then 1.5 years ago or so, he also spent a month out in a remote Aboriginal community teaching science as a volunteer to those kids. And for that work and for the scientific work he's done, he got awarded the Melbourne Rotary Club's Young Achiever of the Year for 2015 recently. Now, one of the things about Jason is for some reason he accumulates nicknames, which is kind of unusual. But I thought I'd share a few with you. So this is Jason at that teaching science to those young Aboriginal kids, and they were calling him this name in the local dialect, which he didn't really understand what it was for a while. And then he found out it actually meant big man girl hair. He wasn't particularly happy about that, as you can explain. So he convinced him instead to call him Big Horse. Now, if you look up Jason on the local intranet, this is what you'll find if you do a search on his name. So firstly, you'll see that he doesn't like using a capital J for his name for some reason. And thanks to Jeff Babin, he's also a good person to ask about bogans and hair conditioner. And pictures like this have inspired other nicknames for Jason, like Jesus, slight resemblance, Rockstar Jason. He seems to think he's a rock star himself in that picture. And then over at the Turf Club, they like to call him Sexy Jace, for the girls behind the bar. This picture's actually when he shaved off half his moustache for some reason, after his brother's wedding, came back, went through the airport and went to the lost property people and asked if anyone had handed in half a moustache. But one of the things about him, besides being tall and hairy, he often walks into your office and asks these questions which don't really make any sense, and then sort of walks out without getting an answer. And for that reason, what most people call him is Chewbacca. It's been great to have around the lab. He's going to give a great talk. It'll make sense, I'm sure. So let's just welcome Jason for his completion ceremony.
Thanks, Sabo. Cool. Okay. Don't really know how to follow that. I'll just talk about what I've been doing during my PhD. So yeah, that's what I'm going to tell you about today is the pro-apoptotic protein Bak and how it goes from being in sort of an inert monomer and then into this larger oligomer that can break open membranes or form pores. I think one thing I wanted to do just quickly before I start is thank the Education Committee and especially Melissa Call, who baked me a vegan cake apparently. What a lovely person. And yeah, just thank them for the opportunity to talk in this sort of format. I think it's better than a division seminar because you have to sort of change your talk. There'll be a lot of structure in this talk, and I'll sort of introduce that as well, so it does make you change it. But, to begin with, apoptosis. So this is what I'm working on. There's two forms of apoptosis. I'm going to be talking about the intrinsic pathway today, but what it is essentially a form of programmed cell death, and it's important for homeostasis and critical during development, and also a defense mechanism against infection. I don't know. Hopefully that doesn't flicker the entire time. Okay, so of the trillions of cells that are in our body, billions of these die every day, and obviously that has to happen in a controlled, ordered manner. Otherwise, you're going to have a lot of problems. So you can see here that they sort of bud when cells are undergoing apoptosis. They bud in a certain way, and I've just got a little video here as well, which is way. I don't know where the cursor. Oh, there it is. Cool. So this is during development. Cells die in between the digits to enable formation of a hand. So that's another thing that apoptosis is important for. This is.
Under the microscope, we can see how cells are destroyed. This process continues throughout our lives as cells become damaged or just worn out. So David Attenborough wasn't available to narrate that, but so why do we care about apoptosis? Because it's such an essential, important mechanism for turning over cells. If it goes haywire, you can get disease, and a very good and well understood, I guess, and well studied example of that is cancer, and that's been well studied at WeHi. But if you have, cells that are dangerous or damaged, not dying, it can lead to the formation of tumours or leukemia and things like that. And the other, probably less well-studied thing that I think Grant Jewson will talk about on Wednesday is when you have an excess amount of cell death. And this has been implicated in neurodegeneration, hearing loss, stroke, and heart disease, but it's not as well established, I guess, as cancer is, with apoptosis being dysregulated. So the intrinsic apoptotic pathway, which I'll be talking about today, is comprised of three members, and I will mainly be talking about Bak and Bax. And so Bak and Bax are these proteins that sit in the outer mitochondrial membrane, and when they get a death stimulus, so something happens in the cell, whether it's DNA damage or ER stress or cytokine deprivation, those things trigger Bak and Bax to be activated and then oligomerize. That oligomerisation somehow forms pores or disrupts the membrane. That releases cytochrome C, other apoptogenic factors that result in caspase activation, and you get cell death. So the way in which Bak and Bax are activated is by the second member, these BH3 only proteins. So they're able to directly activate Bak and Bax and cause those conformational changes that result in membrane disruption. They can also disrupt pro-survival proteins, which are the third member of this group. And they essentially, as their name suggests, exert their effect by stopping Bak and Bax from oligomerizing. They can also interact with BH3 only proteins and prevent them from doing their thing. So if we look at the domain structure, I just want to quickly bring this up, and I'll come back to it later. But the reason why they're in this family is because of these BCL2 homology domains or homology sequences. And there's 4 of these. The Bak and Bax and the pro-survival proteins contain all 4. They also possess 9 alpha helices, and this is quite a structural talk, so I'll come back to that later as well. The BH3 only proteins just have the BH3 domain. So I wanted to sort of represent this a bit more schematically so it's a bit easier to understand for people that don't know, aren't that common with it. So Bak and Bax can exist on the outer mitochondrial membrane. A BH3 only protein can come along, interact transiently with the protein. Oop.
Just keep going.
I think
fix your thing.
This thing isn't working. The pointer is, but the slides aren't.
Just keep going. Seriously, keep going.
Okay. Well, I might. Can I do? Well, I can't. I can't go to the next slide.
All right. What am I doing on the weekend? My girlfriend flew here from Indonesia. That was nice. What's going on? Sorry. Pressure's on, Colin. Sorry.
I'm just getting rid of that flicker.
Okay. And I never had that rockstar Jace name. I didn't name myself that. That's a lie. Most of those other ones are true. Funny you mentioned that, Matt. Well, this thing still isn't working, but maybe I guess I'll just... Oh, it's all there now. Giving it away. Okay, awesome. Thank you. So a BH3-only protein can directly interact with Bak and Bax, and this is a transient interaction. Then the Bak or Bax undergoes conformational changes and can form, well, these dimers, and then dimers can go on and form oligomers that release cytochrome C. And essentially this schematic here is what I'll be talking about today for Bak and Bax. But I just wanted to also put in, the prosurvival proteins are part of the family, so they can come along at this point when Bak or Bax is activated, grab a hold of Bak or Bax, and prevent them from oligomerizing. This is the other point where BH3-only proteins can come and sort of break that apart by binding to a pro-survival protein, and that allows Bak or Bax to form oligomers again. And the final thing is that pro-survival proteins can sort of get to these BH3-only proteins before any of that happens and stop the direct activation of Bak or Bax. And so my PhD is more or less in crystallography mainly, and if we think about the size of these proteins, they're essentially a millionth of a millimetre, roundabout, a little bit bigger than that. And the technique we have to use is some sort of, like you can use cryo-EM or NMR. We're specifically using X-ray crystallography. And so I wanted to just give you a small intro into what crystallography is before I start banging on about squiggly lines and structures and things like that. So what we do is we recombinantly express and purify the protein of interest. In this case, it was mainly Bak. And then we try to get that at a high concentration in a pure form and put it into these crystallization trials. So this is essentially taking thousands of conditions, potentially. They've all got different levels of precipitants and salt and things like that. And it's in a way sort of aimed to slowly bring the protein out of solution and hope that it can stack and form crystals. That's a very sort of rough overview of what it is. And you can do that by vapor diffusion, which sort of increases the concentration of things inside a drop. I won't go into that too much, but if you're lucky, you can potentially get some hits, small crystals or needles or something, and then optimize these into bigger, larger crystals that we actually need to get any data from. So the reason why we need a crystal is also, I guess, reasonably important to explain. So if we shoot, what we actually do is shoot these crystals with X-rays. And if you just shoot a single molecule with an X-ray, the signal is just nowhere near enough. And what a crystal provides is inside one of these crystals, which might be 100 microns across, there could be hundreds of millions of proteins in there, all stacked in a very similar way. And from that, you sort of get an amplification of the signal. So the way that we do this is using the Australian synchrotron. And I'm sure some of you have heard of the synchrotron. I'm a massive fan of it. I love it. It's amazing. It's probably the most interesting scientific instrument I've ever seen. And so I was reading a little bit more about it, and what actually happens is, so there's two rings, an inner and an outer, and this little outlet here is actually an accelerator. So it accelerates electrons up to 99.9% of the speed of light over a meter. So by a meter, they're going that fast. And this ring in here is 130 meters in circumference. And within half a second, that electron, or those electrons, have done 1 million laps of that. So that just, I mean, obviously the speed of light is quite fast. It's just, when I read that, I was like, yeah, that's awesome. So just say it, big it up. So then they get released into this storage ring. They're obviously still traveling close to the speed of light. And when you bend them with magnets, you get something released which is called synchrotron light. It's essentially like a broad spectrum of light, and you can extract, in our case, X-rays out. A super tiny, very intense beam. And so the synchrotron's used for loads of other things, but this is what we use it for. So you grow your crystals, fish them out with these tiny loops, which you pretty much can't have any coffee before you start trying to pick crystals up because your hands shake like that, and that loop's, I think this loop's 100 microns across, so you have to pick them up under the microscope. And then shoot them with X-rays, and when the X-rays pass through the crystal, they interact with the electrons in the atoms of the protein, and they get diffracted off and form this diffraction pattern. And so essentially what you do is you spin the crystal or rotate the crystal as that's happening, and you get something that sort of looks like this, which doesn't play very well, but yeah, so you build up all these different diffraction patterns, and from that, you can generate this 3D map of electron density. And wouldn't have to do it justice, but say that this arrow here represents, I don't know how many years of development and amazing findings. And I mean, I think the person that I know that understands that quite well is Peter Coleman, but I won't go into it because I think I'd be overselling myself to say I understand 50% of it. So it's pretty complicated. We can use a lot of programs, computer programs, to do that. So into your density, you build, essentially take the amino acids that make up proteins and build in your model. You can use a program to refine this and make it more and more accurate until you have a model that looks like this, which is pretty hard to interpret, but you can then represent that in secondary structure. So, for example, this is one of the pro-survival proteins, and the secondary structure is alpha helices or beta sheets. And this is what one of the pro-survival proteins looks like, which is a globule bundle. So this is what we'll be looking at for a lot of the talk, is structures like this. So I just wanted to explain what they actually are. So to come back to this slide, and now we're thinking about these, the family, what do they look like? So the crystal structure of Bak was solved in 2006, and you can see it's sort of, in this case, it doesn't have its transmembrane domain because that disrupts crystallography and expression. But it's essentially an alpha 5 helix surrounded by 7 other helices into this globule bundle. And when you look at a pro-survival protein, in this case, BCL-XL without its transmembrane domain, you can see how amazingly similar they are, remembering that this thing kills the cell and this thing keeps it alive. And I think that was a super interesting question when I came to the lab. I'm fascinated by that straight away. So I'll talk a little bit more about that later, but these are the 2 proteins that I'm talking about today. So this is the NMR structure of Bax with its transmembrane domain actually bound back to itself. And I wanted to just mention this here because Bax can actually sit in the cytosol or at the membrane, whereas Bak is predominantly at the membrane. So these 2 proteins will be essentially the main focus of the talk. I also just want to mention that there is a hydrophobic groove where BH3 domains of other members can bind. So I guess, the important point to take away is that they all look quite similar and they all contain this hydrophobic groove. And as I said, we'll be talking about this direct activation. So to begin with, I think I had to introduce this by talking about the work that's been done in the lab before me, and that was by Peter and Dana on Bax. And my project follows along with Bak and understanding activation of Bak, and initially Bax was sort of paved the way for a lot of my work. So the first thing that they did was take a recombinant form of Bax, lacking its transmembrane domain, incubate it with this BH3 only protein BIM, just a peptide of that, and chaps, or a detergent, a non-ionic detergent. And they saw the formation of this dimer. I'm not going to go into why OM does this, but the BIM and chaps we're just as interested in. But so what they actually did was take this dimer peak and put it into crystal trays, and solved this crystal structure. And if you remember, the monomer I just showed has this alpha 6, 7, and 8 actually coming back up here. So this is a structure of a domain-swapped dimer in which, using BIM and SHAPS, this alpha 6, 7, and 8 has come out and gone inserted back into a corresponding molecule. So what does this mean? Well, so when they first saw this, they thought, okay, so when you activate Bax, it separates into these two domains, a core and a latch domain, and sort of depicted here by this latch coming out. Now, this has sort of been one of the things that people have misinterpreted from this paper, and from my work as well, is that we don't actually think, and they didn't think, that this dimer is physiologically relevant, but the conformational change that happens to allow it to crystallize like this could be. So to prove, or to try and prove that this alpha 5, 6 separation and the latch coming out was actually important for cell death, they inserted two cysteines into the alpha 5 and the alpha 6, and then you induce this to disulfide link these two residues, and that should form something that blocks the latch from coming out. And that would be reasonable proof that this conformational change was important. And that's exactly what they were able to do. And here, so this is using double knockout Bak and Bak cells, and you reconstitute in this double cysteine mutant, and then isolate the mitochondria, and essentially do an experiment looking for cytochrome C release. And so when you add TBID, or the BH3 only protein, you see cytochrome C release, which means the mitochondria is broken open or has pores in it. Whereas if you put this QFE oxidizing agent, which induces this disulfide link in beforehand, you block that. So from this, they're now thinking that Bax can exist, or when it gets activated, this is sort of the active form of Bax. So there's a core domain and a latch domain, and they separate when a BH3 only protein directly interacts. So one other thing they were able to do with that structure of that dimer was to supplement in excess BH3 only proteins, in this case BID. And when they did that, so having a 5-10-fold molar excess before they put it into crystal trays, they saw they could solve a structure with BID bound to each end of this domain-swapped dimer. And I think you'd look at this and think, okay, it's quite artificial. I mean, crystallography is, especially with recombinant proteins and things like that. But they were able to go through and mutate residues and then show in cells that the crystal structure they were looking at did make sense, and it was physiologically relevant. So it sort of was building up this model of what's happening to Bax during activation, that the BH3 only protein can bind in the hydrophobic groove and then result in these conformational changes. So the next thing that they wanted to understand is what does this dimer or oligomer look like? And the reason I say dimer initially here is because a lot of the work that Grant and Ruth have done sort of characterising a BH3 and groove dimer. And you'll notice the way that I've drawn this schematic is the little orange bit here, which is actually the BH3 domain, is inserting into another molecule. So there was already quite a bit of evidence for a BH3 groove dimer, sort of like a symmetric dimer, but we only had models for that, no structural evidence. And so Peter and Dana were also able to, I'll just mention that, so that's what I was just talking about there. There was also this other study that found that the alpha 2, which is the BH3 domain, which was the orange part that I was talking about, and the hydrophobic groove, the alpha 3 to 5, plus a transmembrane domain, that small segment, they were suggesting that that was all that was required for membrane permeabilization. And I'm not convinced. Grant tried to reproduce these experiments and not had much luck, but at the end of the day, they rationalized the design of this construct sort of based on this as well. So what Peter did was to take this alpha 2 to 5 and attach a GFP molecule to it. So it would be quite hydrophobic, so that was to solubilize it and potentially aid in crystallization, and purified what looked like a tetramer on gel filtration. He also made other constructs, but the one that worked was the alpha 2 to 5, and was able to solve the crystal structure of that. So at the top here is 2 GFP molecules, and these exist in dimers, and then between them, this is the part we're interested in, are 2 BH3 groove dimers, or core domain dimers is what I'm going to call them. So what does that look like? So before you remember I was talking about core and latch separating. Well, this is essentially the core domain without the alpha-1. And you can see that the BH3 domain of one molecule sits in the hydrophobic groove, which is the 3, 4, 5 of another molecule, and it creates this symmetric interaction. One of the other interesting things they saw was this sheet or face of aromatic residues that lined the bottom of the dimer, and they thought this could potentially be important for membrane interactions, considering this thing does somehow permeabilize membranes. So this was sort of building up this model again, and you can see here what I was talking about. It's sort of, you can come from the core latch separation, it's consistent with the formation of something like this BH3 groove dimer, which has been pretty extensively shown in cross-linking experiments to be what's happening in a cell as well. But you can see that there's still quite a bit of the protein left here. So this latch domain's gone from this construct, so is the transmembrane domain. So we sort of think of this core domain as sitting at the heart of this BH3 groove dimer, which is a bigger dimer. But what happens next is still a little bit anybody's guess. So yeah, I mean, and that's what I'm going to talk about a bit more with Bak later. So what about Bak? So this is where I guess I came in and started doing experiments. And when we look at what are the differences between Bak and Bax, or why, I mean, a lot of people think that they're functionally redundant. And the reason they think that is because if you take single knockout Bak or single knockout Bax, there's very limited abnormalities in their phenotype. If you knock both of them out, then nearly all the mice die in litter. So I guess we know, at this point, we knew quite a bit about Bax, and nothing really about, not too much about Bak structurally anyway. So I kind of thought this was a perfect metaphor. So this is Bax here, and we sort of know what that looks like, and then we're just copy-pasting that, what we think it looks like, onto Bak, and you get lost a bit. So anyway, what we're trying to do is figure out what was going on with Bak. And the first thing we did was to look at the differences. So Bak is predominantly the membrane, as I said before, whereas Bax can be in the cytosol or the membrane. Now this is quite contentious, but there's been reports that suggest that varying responses to BH3-only proteins. However, I'd be more inclined to believe Colin's work that was recently published that suggests that that's not the case. There's also, Bak has one activation site, likely only has one, whereas Bax potentially has two. And there's also these interactions that could occur before these proteins are activated. And it's quite well characterized for VDAC2 and Bak, that interaction, but as for what is interacting with Bax is still, I think, quite unclear. So what do we know about Bak activation? We know that the crystal structure of the monomer looks like this. We also, there was an NMR structure that came out about halfway through my PhD showing a stapled form of a BID peptide, which is reasonably artificial, bound to the hydrophobic groove of Bak. So this is an NMR structure, and it's quite hard to see the specific details in it, but one thing that was very clear is that it looked like the BH3 domain bound in the groove. As for structural changes next, we knew the BH3 domain became exposed, and we also knew that the alpha-1 at the back here got displaced. But structurally, we weren't really sure if the core and latch separation could happen as it does in Bax. And as I said before, a lot of work had been done on a BH3 and groove dimer forming by Grant, but we weren't sure what that structurally looked like either. And as I said, the oligomer was, quite, it's a huge thing, and I think it's still relatively unclear what's going on with that. So the first thing I did was sort of, follow along the lines of Bax and throw all the BH3 peptides that I had, plus, this zwitteronic detergent chaps, and I can go into more what we think that's not doing, but it's not quite clear, but potentially aiding in the dimerization or activation. And I took this peak here, the dimer, and put that into crystal trays. And you can't see very well, but there's some sort of smallish thin plates down here which diffracted quite well, and we were able to solve the structure of a domain swap dimer of Bak. And you can see straight away that this alpha 6, 7, and 8 has come out away from the rest of the molecule and inserted into a corresponding molecule in the same way that was shown for Bax. So we're thinking about Bak in the same way, that it separates into this core and latch domain through this sort of conformational change. And we wanted to go on and show in cells, what was actually going on. So to prevent this conformational change from happening again, I scanned a few different cysteine mutants and came up with these two, which seemed to be the most efficient at cross-linking and could still kill in a cell when they weren't cross-linked. So that's the F150V142 mutant. And did this experiment in the same way with double knockout MEF cells, and then you reconstitute this double cysteine mutant back in and then do your experiments. And all this work that I've done in cells was with Ruth Clark. And everybody in that lab was wonderful. It was a great place to be when I was doing these experiments. So what we saw was that if you look at the single cysteine mutants, and the double cysteine mutant, and you just add TBID, which is the activating BH3-only protein, you see cytochrome C release in all of these. So they all still look like they're functional and they can kill. And I'll disrupt the membrane. And if you add QFI in, induce this disulfide link with this oxidizing agent, the single mutants still release cytochrome C, whereas the double mutant blocks. So from that, we sort of came to the same conclusion that tethering these two helices together prevents cytochrome C release, and convincingly that this conformational change was important. So in the same way as was for Bax, we were thinking about this as the active form of Bak, where the core and latch are separated. So I also did some experiments similar to Bax with supplementing on excess peptides, and I tried essentially every peptide we thought that would bind to Bak, and lots of different truncations and stuff like that, and this one was the one that worked and worked beautifully. We got nice big sort of cubic crystals, and I was able to solve the structure of that to quite a high resolution. So you can see it in the same way, it's got two peptides, one bound at either end. And this is actually what that interaction looks like, and I haven't shown this for anything else, but I'll just quickly tell you that these four or five, potentially 6 hydrophobic moieties poke into that hydrophobic groove, and this is sort of what drives this interaction, as well as a salt bridge over here. And those sort of characteristics are what is seen in all of these BH3 groove interactions, so as well for Bax. But I'm not going to go into that too much. So this was important, and this is something that we haven't published yet, but it will add to the literature because it is, first of all, BIM instead of BID, which is potentially interesting, but this is quite a high resolution structure, and we can see exactly what every side chain is doing, which is aiding us in drug development. We can also, I think it was also important that this peptide didn't have a staple in it, whereas this one did, so it was a bit more native. So next is the same sort of question, what does this core domain look like? So the core and latch domains are separated. Can this as well go on and form a core domain dimer? So I created a construct that was similar to what was done for Bax, and then tried to crystallize this. It purified as the same sort of size tetramer, and thinking that the GFP might be driving a lot of the crystallization, I tried all the different conditions that were done for Bax, but didn't get much. And interestingly, this alpha 2 to 5 of Bax we thought would be quite hydrophobic. It actually crystallized in the presence of the detergent chaps. So I decided to try and screen a few detergents and see what happened. And then this is a Blue Native page, which allows the interactions between proteins to stick together on a gel, essentially. And you can see that it wasn't really happy to begin with, the Bak construct. And as we add in all these different detergents, non-ionic detergents and zwitterionic detergents, some of them did make the thing much more homogeneous. And I set up most of these in crystal trials. The two that I got crystals from are these two here, OG and C12E8. And you can see these crystals are pretty cool. They're green because of the GFP. And was lucky enough to collect a good enough data set to solve this structure, which is the tetramer of GFP and the Bak alpha 2 to 5. And you can see a dimer of GFP at the top and the bottom, and then in between are these two dimers, sort of exactly as we expected to see the BH3 groove dimers. So if we take out one of those, we can see that it looks quite similar and essentially what we'd expect, with a BH3 domain sitting in the hydrophobic groove of a corresponding molecule, creating this symmetric dimer. And just to make sure that this is what was an accurate representation of what was in a cell, I did some cross-linking experiments where I mutated either this isoleucine, aspartic acid, or arginine to cysteines along the dyad axis. And these two we should be able to link because they're close enough with this BMOE linker, which is about 8 to 10 angstroms, but the I80 would be too far away. So when we induce oligomers with TBID, we can see that we could link with this cross-linker dimers after activation. This was convincing that what we saw here in the crystal structure was in fact in the oligomer. We also saw this aromatic face again that was seen for Bax. And when we compare that to Bax, you can see that, you see what I'm talking about. So Bak is a bit more curved. We're not really sure if that's from the GFP proteins on either side or that it is actually relevant. But the point I want to make here is this has been a bit contentious. I think even Doug Hilton asked a question last time Peter talked, like, is this reasonable to think that this could be, something how it associates with the membrane? Other proteins have been seen to have this, and this is one example here, which is a dehydrogenase that sits in, I think, the inner mitochondrial membrane. However, yeah, I mean, it's still, I think we'd have to do mutagenesis studies and things like that to really understand whether this is important. So this is in the same way as it was done for Bax. We're sort of building this model of what Bak activation looks like. And in the same way as before, we're thinking about this core domain dimer as lying at the heart of the bigger BH3 groove dimer. But what happened next, we're not really sure. So one of the things that this crystal structure provided was these interesting crystal contacts. And in the beginning, I was talking about, a protein builds sort of, in a repeating fashion, you get crystal contacts between monomers or in this case, a tetramer in a crystal. And we saw something interesting in this crystal packing that could be involved or, hint at what an oligomer of Bak would look like. And this is essentially if you take a part of the crystal out, what we were looking at is, so inside this tetramer, you can see this interaction, which is the alpha-4,5 loop. And that interaction was exactly the same as between tetramers. So it sort of formed this ladder through the crystal. And we got pretty excited by this and thought, well, is this how Bak is killing? So we came up with some pretty crazy or idealistic models, and Brian helped us with this. And this is actually Bax, but you could think about how potentially this thing could sort of wrap around, and this is the hydrophobic aromatic residues around the outside and hydrophilic inside, and potentially form some sort of pore. You'd always have to test this because it's obviously quite a little bit outlandish. So what I went on to do then was to look at this interface and see if I could link things across here, and was that feasible to be making that sort of model in the first place. What I saw was no. So there was pretty much nothing happening there. Occasionally we would get some linkage. So this is just a reducing SDS page gel looking at, in the same way before, could I link dimers in an oligomer? And the answer was essentially no. And this wasn't surprising considering we were dealing with a crystal contact, which is essentially very artificial in the first place. But it does show you that crystallography can give you these extra things that you wouldn't usually think about. And there's lots of publications showing that the way a crystal grows can be physiologically relevant if something was going to oligomerize in a cell. And this is also just a death assay to show that all the cysteine mutants that we make Bak can still function the way it should in those. So we didn't really get any answers from chasing that. It was a bit of a rainbow to chase, I think, but you've got to do those things just to make sure that's nothing. And what we do know, so yeah, I mean, we don't really know what's going on with the oligomer, but if I try and come back and hypothesize or model what this thing might look like outside of the BH3 groove, or the core domain dimer. So if we look at this, we know we've got a structure of this part of the groove dimer, but the latch domain we know comes out, and the alpha-9 is attached to that, and the alpha-1s come off somewhere as well. So I guess the way to try and understand what this thing looks like is going to be quite hard through structural biology, because we think this is sort of more like maybe a solid part of this oligomer, but these are quite soft, and we're not sure if that's quite flexible, and chasing an interface could be quite hard. So what we do know is that you can link residues on the alpha-6, and that was shown by Grant. You can also link residues on the alpha-3 and the alpha-5, and that was some of our work and this Alavila study as well in mouse back. And Shweta recently, her recent published work is showing alpha-9. And Ruth Clark's lab does a lot of work on sort of scanning, putting cysteines in positions and trying to understand what this oligomer looks like. And I think that is one of the best ways to try and figure this out, because it's essentially like spaghetti and one meatball in the middle. It's pretty hard to know what's going to happen and how they're going to interact. And this is me just sort of maybe giving an idea how it might sit on a membrane initially. So now I guess we're thinking about sort of bringing this all together. We're thinking about Bak and Bax as a combined, having a combined model of activation. So initially they sit in these inert sort of monomeric states. Bax can be the cytosol of the mitochondria. BH3 only proteins can come in and bind through this hydrophobic groove. And these structures are really, I mean, as Peter sort of suggested in the intro, we're now using these things for trying to design and develop drugs. And so we brought in the big guns to help with that. This is someone explaining to Peter how to design a drug. And Davo's not listening. So after that, after this BH3 and groove interaction, the Bak and Bax can then open up and be activated in the same sort of way, and we think that's the core and latch domains separating. And if you remember at the start, I was talking about pro-survival proteins and Bak and Bax having this very similar fold, and how I was quite interested by that. So one of the things that we're thinking now is obviously the conformational changes that happen after a BH3 protein binds. So we know that for Bak and Bax, this is a transient interaction, whereas for pro-survival proteins, essentially they stick and hold the BH3 only protein. So this is potentially one of the reasons why they look similar, but pro-survival proteins may not be able to undergo this conformational change. And it's worth me pointing out that this has been seen for BCL-XL, but not in a physiological setting. So yeah, I mean, I think that's still up for debate whether pro-survival proteins can do that or not. But it could be a way that they look the same, but are functionally very different. So then they can go on and form this interface, which seems, quite solid and is potentially a building block for the oligomer of Bak and Bax. But as I said, there's quite a lot of work that's been done and not that much that really convinces you what these things look like. And that may be because membranes are also involved in these interactions between BH3 groove dimers, which makes it very tricky to study. So if anybody is asleep or has been asleep or, I don't know, dozed off or thought about something else, luckily you can still take something away from this talk because I made a really sweet video. So usually I'd apologise to Peter Coleman for these sorts of things, but he told me he likes it, apparently. Okay. Oh.
Baby got back.
So obviously I have to thank a fair few people. And no bigger than anybody else, of course. The biggest influences for all this work have been the Peters, Little Peter and Big Peter. It's not by size. But yeah, so these two guys, I guess when I first came here, I instantly knew this is where I looked at maybe 10 labs for my PhD, and I knew straight away within 5 minutes of talking to them that we could work well together. You know, they're fantastic scientists, and I think most of the people at WeHi know that. But they're also great people. They've been mentors to me, not for just science. They're, you know, in life as well. And I don't know, I feel very lucky to have been in their lab. And hopefully we'll be working with them a little bit longer before I go. I'd also like to thank both the Coleman and Zavatar labs. It's been an amazing place to work, and I've had, you know, had a really good time during my PhD. And yeah, I mean, I think that has to be said for the division as well, and even bigger, all of WeHi. Like I love this place. It's been awesome to be here. I especially want to just say a shout out to Doug and Arena. I've worked on a bit of other stuff with them that I haven't talked about today. Also Babalap, he is a mentor and a friend, and he's always got good suggestions. Callum helped me a little bit with that video, but he sort of more didn't help me. Like he led me in wrong directions, and I think it took me longer. Anyway, for his enthusiasm, I can rip on him because he's in Japan, so it's fine. Cyrus, he's usually got some witty thing to say, or a title, but anyway, yeah, he's been great as well. All the students in structural biology, our dumpling crew, have been great. Also want to just acknowledge Ruth and Grant. I've done a lot of the cell work that we've done together. They've been great, and the MGC division and their labs is wonderful as well. That's a great division. I would have been, happy to be working there. There's a lot of lovely people there. I originally did some work with the Davids, and Guillaume is helping us develop these drugs now. And it goes without saying that most of this stuff wouldn't have happened without C3. And this is a crystallization aiding facility where they crystallize your proteins essentially in an automated way, and Janet and Shane have been wonderful with that. Brian Smith helped with some modelling, not just the Pore, lots of other things as well. He's been great. My committee has been fabulous as well, especially Matt Call for his great announcements about interrogating me, and he's bought me a few beers over the years as well, which is good. Yeah, I'd like to also just thank the Reconciliation Committee and it's been a fantastic committee to be part of, because we're actually getting things done. We're doing things. I feel like it's progressed quite a long way in the 2 years that I've been doing it, and yeah, it's great to see. And I want to just send a shout out to Louise as well. She's the head of that. She's awesome. And I think bigger than everybody, the support network that I have is from my friends and family, and my girlfriend flew from Indonesia just to watch this talk. So that's amazing, and obviously thank her a lot. And my mum, who's been there from before day zero, she's always given me everything that I needed, and she's a wonderful mum. Yeah, and to everybody else, thanks for listening. Thanks for coming.
Right, got some time for questions, so shoot away. Okay. Ruth, go first. Sorry.
The structures we've got GFP bound to the core domain look a little different from Bax to Bak. So the dimerization between the GFP domain is different, or can you explain how those structures turned out?
Yeah. So I don't have a good picture comparing those directly, but so one of the things is the crystal contacts are different. So you could see that the way that Bak was a ladder through the crystal, that didn't happen in Bax. That's one reason.
So do you think that was driving it? Or if it was driving it, you might expect it to drive the pores, but you didn't see that.
Yeah. So, it may just be a reasonably weak interaction that when you get proteins at 50 mg per mil in a crystallization condition, they come together in that, that's an energetically favourable way to come together. The other thing is that Bax structure is quite low resolution. So the way that it's formed, I guess it looks more flat instead of so curved. And that could be, based on the whole, I guess that core dimer is more flexible, and that's why the resolution is quite good with the GFPs. But yeah, if you think about the crystal context that was seen for Bak, sort of held Bak in maybe stabilised it. Those crystal contacts aren't there for Bax. I think it's hard to compare a structure that's 2 angstroms and one that's essentially 4. So, but they could be different. It's hard to tell at that point, I think.
Anisha?
Jason, great talk.
Thanks.
I might have missed this, but I just want to know, when you do your in vitro dimerization experiment, you always have a detergent, is that right?
Yeah.
Because the flip. So what in the cells, what's in the cells, like how is, like in the cells obviously detergent's not there. So what do you think is a factor in the cells that causing, and why do you think if you're doing it in vitro, why is it not capable of causing the flips on its own without requiring detergent?
Yeah. So it's something that we're still not 100% sure about, but the detergent is potentially, we know that CHAPS, which is the detergent which I use in that assay, does have some affinity for Bak. So it may be binding in the groove or somewhere else on the protein, which sort of destabilizes it enough or enables it to be activated by peptides. The reason why I think comparison of in vitro to in vivo is Bak is at a membrane. We don't know exactly, the membrane's quite complicated, it has a lot of different things in it, the outer mitochondrial membrane. So we don't know exactly what it's mimicking, that CHAPS, but it seems to be, it's potentially trying to mimic what a membrane environment could be like. It's obviously very different and artificial, but that could be one of the reasons why it needs to be there. But we're essentially not 100% sure.
Right.
So first, thanks for giving me something to live up to on Wednesday. So you talked about obviously the different inflections of the Bax and Bak monomers, the Bax dimers, being flat and the Bak dimer being curved. Given that they can co-associate seemingly in a cell, which do you think is the most accurate reflection of what's going on, or how are you going to test it?
Yeah, I mean, and like you say, so those things can heterodimerize as well, which I didn't say. But yeah, I think it's very hard to know. Given that the crystal structure is attached to a GFP which drives other contacts, and that curvature might be a result of that being sort of forced together. You don't see that in Bax. I'm not sure, because when we think about it, we don't really know how those Bak and Bax dimer would interact anyway. So it's quite hard to say. But maybe there's some, it's on a membrane which is fluid to some degree, so there could be any number of conformations like that it could adopt. So the answer is I don't know, but...
I'm sure you've looked at it, but is there any avenue for making loss of function mutations in that region which I guess you have to worry about causing disruption of the actual protein structure itself?
Yeah, so that's something that I haven't done, but I think Peter's thought about this, and I think I guess it's hard because if you think about those aromatics that are on the membrane at that point, they were initially probably inside the protein, and those things are usually key for stability. And if you mutate one of those big hydrophobic aromatic residues, like you say, maybe you know better than anybody what's loss of function in Bak, but I'd say it's going to be challenging, but maybe there is a mutant that won't be at the mitochondria when it's an oligomer. I don't know.
Jason, so the cysteine cross-link on the alpha 5-6 core latch experiment, so I think those are really elegant experiments, but just to play devil's advocate, the functional readout on those experiments, if I got it right, is always cytochrome C release upon TBID treatment. Can you rule out that disulfide bond just blocks the TBID interaction and not necessarily the core latch transition in response to a TBID interaction?
Yeah, that is a good point. I know, so I didn't show the data, but for example, for Bax, when you actually tether that, I don't know if Grant's done this experiment in cells, but you actually increase the affinity. It actually locks BID in there. So it's potentially, I guess there's a potential for that, maybe not the interaction stopping it, but it making it so it can't come away. But I don't think we haven't done the experiment to specifically rule that out. But it is a good point.
Lisa.
I have a kind of fairyland question for you.
Yes.
Assuming that if we go with the Pore model, assuming that they come in and they kind of oligomerize then in a circle, what do you think would be the mechanism for actually removing the lipids in the center of that? So it's not just making a ring with lipids in the middle. How could you imagine that actually that oligomerization would cause the lipids in the center to be displaced?
Yeah, that's a good question. I personally don't necessarily think it's a pore, but to answer your question, I think there are a few models where you can think about, half a ring forming and then the membrane being destabilized. It might not have to be a full ring. I only show that because it was a model that was easy to sort of understand why you would chase it. But in my opinion, what's in the literature, a pore can take on many meanings, and sort of we've had quite a few discussions, Peter Coleman and Peter Zavazar and I, that if all these things, the last sort of spaghetti thing that I showed, if they could just all have low sort of reasonable affinities, but all just come together in different ways and sort of cluster, and then we don't know if that could disrupt the membrane. I think it was called a sinking raft model or something like that. So that's another way that it could happen. It doesn't necessarily, and that would essentially create a whole cytochrome C would come out. I don't know if you'd call that a pore, but I think I'm not convinced by it being a circle. So yeah, I mean, it's a good point. It's a good reason why you might not think that it is a complete circle like that.
We'll have one last question from Jeff up the back.
Between you guys and Ruth and Grant, you must have done a pretty extensive cysteine scan of Bak and Bax. So are there any cysteine mutants that you can make that do cross-link an oligomer? So you were looking for someone to validate your model informed by the crystal pack, you didn't find any. Are there any stray cysteines, stray residues that when you put it in a cysteine you do start to see them cross-linking together?
Yeah, so I think what you're talking about has been so lots of these things have been done. For example, Grant published a paper in 2009 where he saw that in the alpha 6, but they're never residues that go bang, and that's all the cross links. And I don't know if I don't think Grant would convincingly say that it's just the alpha 6 that's needed for that interaction. There's another paper some of our work had the alpha 3 linking, and there's another study with mouse back that has the end of the alpha 5 and the alpha 3, and Schwetter's work, which looks at the alpha 9, and I think also Ruth's done quite a bit of stuff with the alpha 1 linking that I'm not exactly sure exactly what's been done. Those guys would have, I'm sure that Ruth has a pretty good idea in her head what links, what doesn't, and how it might come together. But I think the answer is there's multiple answers, and it's hard to sort of think about how all that information can fit just one model.
Right, and with that, it just remains us to thank Jason for an interesting and entertaining talk.